So our next speaker will be Dr. Maier, and Dr. Maier will introduce you. Feel free to start sharing your slides. Dr. Maier is a biochemist and infection biologist by training. She received her PhD from the Institute of Microbiology at the ETH Zurich in 2014. During her postdoc in the laboratory of Nassos Tipas at the EMBL Heidelberg, she developed high-throughput methods for the systematic investigation of drug microbiome interactions. Since 2019, she has been leading her independent research group at the University Hospital of Tübingen, where she was recently appointed to a full professorship. In her lab, she investigates whether drugs can be used to specifically modulate the human microbiome. And with that, yeah, please take it away, Dr. Maier.
Okay, thank you very much for the kind introduction. Can you hear me?
Yes.
Slides are okay?
Yeah, we can see it. There is like some slide overview at the left. Maybe we're in the speaker view, I'm not sure.
So maybe I can.
Maybe if you try to put them on full screen, it should show up.
Okay, let me quickly try again. Better now?
That looks great, yes.
Okay, sorry for this. Okay, thank you very much for the kind introduction. Thank you very much for the invitation to speak. Yeah, so I'm speaking here from Germany, so we are about to have we're close to 7 o'clock, so if it gets darker and I have to switch on the light, please excuse me for a few seconds. Okay, so the title of my talk, Systematically Dissecting the Interactions Between Drugs and the Human Gut Microbiome. So I want to start off with a couple of introductory slides on the interaction between drugs and the gut microbiome in general, and then I will take it to what we are currently doing in the lab, some things that we already published, what we are interested in. But I want to really start with a broad introduction to possible interactions between drugs and the human gut microbiome. I think it's exciting times at the moment because we really begin to understand that if we are about to understand differences or interindividual differences in responses to drugs, that it's maybe not enough to just look at the host, at the human genome and how variation in the human genome influences drug responses. We are not alone. There are many additional genes and microbes that colonize us, and they actually can be part of the explanation why certain drugs work better in certain patients and have stronger side effects than others. So it's really the time now to not only focus on the host genome, but also look into the many of the trillion of microbial genes that are there and the strong interpersonal variation that they bring on the table as well. And overall, to get us started, I wanted to give an introduction on possible interactions, and broadly speaking, can be subdivided into two types of interactions. On the one hand side, we have direct interactions that are host independent, and we have indirect interactions that are actually depending on the host. And in both cases, they can go in both directions. So drugs on the microbiome, microbes on the drugs. So I really would like to guide you through what we already know, explain a little bit why it's so difficult, and then also show ways how we actually can tackle these complexities. Okay, so let's get started with the direct interactions, drugs on the gut microbiome. This is something that has been shown a while ago already, that if you ask the question, what explains most of interindividual variation in the gut microbiome composition, the answer is pretty clear. It's medication, the drugs that people take. And that might be pretty clear for antibiotics. Of course, if you take an antibiotic, you have a strong collateral damage on your gut microbiome. That's clear. Antimicrobial effects, this has been known for a very long time, but also can imagine that certain drugs might favor the bloom of certain species within the gut microbiome. So you can have compositional and functional consequences of the direct effects of drugs on the gut microbiome composition. As mentioned, very well known for the antibiotics, we have the collateral damage, we have side effects, we have associated diarrhea. I will show you in a couple of minutes that this also happens a lot for non-antibiotic drugs. And there it's a little bit more intriguing, but also quite interesting, because we have first hints that it's not only an antibacterial effect, but these interactions can be part of the mode of action of the drug, or it can explain side effects. Now, the best or the poster boy example in the field, and that's the first drug that we realize that also non-antibiotic drugs can have an impact, is actually metformin. So metformin is a drug that is used for type 2 diabetes. And what has been shown very early on in the field is that there is a difference in the gut microbiome composition between healthy individuals and patients that suffer from type 2 diabetes. And the conclusion was, okay, yeah, type 2 diabetes must result in a change in the microbiome composition. However, it turned out that the result was confounded because many of the patients actually took the anti-diabetic drug metformin. So metformin is a drug that you really have grams of metformin, so really high dosages in your large intestine as well. And it turns out if you have a treatment-naive type 2 diabetic person that starts treatment, that just starting treatment with metformin is enough to change the relative abundance of more than 80 bacterial taxa. So it's not an antibacterial drug, but there is a clear effect on gut commensals. So why do I think that can be part of the mode of action of the drug? So what has been then shown is if you put metformin in a way so that it's just released in the large intestine, that is enough to improve glucose tolerance. So that's basically an indication that the site of action is also the intestine, because if there more of it is there, that improves kind of the therapeutic effect. We also know that if you take a fecal sample from a patient that has been treated with metformin and you put it in a mouse, just transferring the fecal sample is enough to improve glucose tolerance. So a sample that has been exposed to metformin without metformin being present in the mouse improves therapeutic effects. How this is the case, we do not actually understand. One hypothesis, one possible explanation is that the treatment reduces Bacteroides fragilis, which encodes a bile salt hydrolase that results in a higher abundance of a certain bile acid, which then antagonizes FXR signaling, which is the master regulator on the host side for glucose and lipid metabolism. So here, this is an example where we have like a first idea that the interactions of drugs with the gut microbiome can be part of the mode of action of a drug. Okay, so next type of interaction, again, the direct interaction, microbes on drugs. And there again, we have several possible effects. First is a bioaccumulation effect. That means that the drug is just intracellularly stored within these microbes, thereby reducing bioavailability. We can also have biotransformation. That means that the drug is modified into a metabolite that can either be an active metabolite. So then in the presence of a microbiome, of microbes, you would have increased bioavailability. It can be inactivation, then you would have reduced bioavailability, or the microbes can convert it into a toxic product, then you would have basically side effects. And we have, for all of those, we have a handful of examples that we know also for a longer time. So this actually happens, but we have more like anecdotal examples for these kind of interactions. So when we looked into when you look into what these drug transformation actually do, it's mainly reductions in hydrolysis. This is basically explaining metabolic needs of these microbes within the intestine. So reduction basically would result in increased range of alternative electron acceptors for anaerobic respiration, and hydrolysis would also provide substrates for microbial growth. In most cases, we actually do not know the enzymes that are responsible for these reactions. Okay, so let's move on. Let's look at the indirect interactions. So those are all host dependent. Again, it can go in both directions: drugs, microbes on drugs. And the first example is here: you take a drug and that changes host physiology. I mean, that's the idea of a drug. It's active compounds that are supposed to do something with your body. So if physiology is changed, that again can then impact the composition of the microbiome. And a good example in this context are, for example, proton pump inhibitors. They will increase the stomach pH. That means what can colonize the gut is differently. So you have a change in a microbiome composition. But also we have the other way around. Depending on the gut microbiome composition, host physiology can be influenced. An example is here: change in gene expression. host genes that are important for drug transport, host drug metabolism, and that of course result in different efficacy of a certain drug. But you can also have direct interference with enzymatic activity of certain enzymes that are responsible for drug metabolism. So an example here would be pegresol, which is a derivative of the gut microbiome, and it competes with acetaminophen for sulfotransferases. So if there is high levels of pegresol, it's more difficult to convert an active acetaminophen to an inactive one, meaning it's longer active, and that changes, of course, the efficacy of the drug. And of course, we also know that the gut microbiome trains our immune system, modulates immune processes, and if the drug's mode of action depend on your immune system, of course, the microbiome can possibly interfere with these processes. So good examples here are immune checkpoint inhibitors, CAR T-cell therapy can also be microbiome dependent. Okay. So I think I've showed you a few examples of interactions between drugs and microbes that we already know of. That, of course, directly has implications for therapy. So, for example, you can imagine that if you already know the strain that would degrade your drug, I mean, let's just inhibit it by a co-therapy and inhibitor. If it's a certain strain, you want to target the strain, or you can inhibit a particular enzyme that is degrading your drug. You can also use that as biomarkers. So if I know the enzyme for drug degradation, and if it's there, I maybe adapt dose, or I directly give another drug. So that can help for therapy as well. Another aspect is, of course, that the gut microbiome can produce drugs as well, and we can mine those. And yeah, what I'm mainly interested in is seeing whether we can also use drugs to modulate the composition of the gut microbiome towards a more healthy or beneficial composition. Okay, so now moving more to the general introduction to our specific research in my lab. So most of what we know between the interaction or on the interactions of drugs and the gut microbiome is based on large population studies, and they are awesome. I think you also learned to handle the data that comes out of those in the last couple of days. So, I mean, they are great, and I like them because they give us direct clinical relevance. We know that things are happening in humans. However, in the patients, all these types of interactions that I've just told you, the direct one, the indirect one, this direction, the other direction, happen at the same time. And that's can be a little bit confusing because you just see the outcome and you cannot disentangle. Is it direct? Is it indirect? What are the enzymes and so on? So if you want to identify the causal paths, the interactions and the underlying mechanism, it can be extremely helpful to go to take a different approach. And this is actually what we do in my lab. What we do is to really take a bottom-up approach, a culture-based approach to microbiome research. So we look into drug microbiome interactions in pure culture, then we go stepwise, more complex, so define communities that are derived from human stool. We have host cell readouts, but we also then check in chnotobiotic animals and then compare to cohort studies. And this is exactly what I'm going to present in the next couple of minutes, this whole bottom-up approach to drug microbiome interactions. Now, we know now that many of these microbes are anaerobic bacteria, and we have many, many different species in our intestines, and we have a lot of drugs on the market. So this is just possible if we have high-throughput assays, and we have these high-throughput assays by putting robotics inside anaerobic chambers so that we have scalable and automatable assays, and we have different assays running now in the lab. Most of the time, we just look in bacterial growth, so we have growth assays. Can also be more complicated, relative abundances, of course, if we look into a community setup. So towards a little bit more introducing my own research question. So I've showed you for this drug microbiome interaction, these anecdotal examples for these interactions for few drugs that we knew of. But what I really wanted to have when I started off as a postdoc at EMBL in Heidelberg in Nassus Tüpper's lab was a systematic overview and systematic map on all possible interactions that can take place. Wanted to know whether these effects are direct, are they depending on just one microbe? Is this an effect that just emerges in a community? Is this effect host dependent, and what are the underlying molecular players? Again, we have both directions. So drug degradation, bioaccumulation, transformation. It's not the main focus of my work. However, still I want to emphasize that this has been done now as well. So here we also have systematic or more systematic maps now. And the current estimates are that 2/3 of the drugs are actually metabolized microbially. We will have to see what's the consequences for the host. But yeah, I mean, it is not these few examples that I just introduced, but it's really many. It's rather, it's the rule and not the exception. Now, I'm more interested in the direct effects in terms of growth of a variety of different drugs. So this is now the topic for the next couple of slides. Okay, when I started off my postdoc, I wanted to have the systematic map. So we started off selecting 40 species of the healthy human gut microbiome. They cover quite a high percentage of the genera that are present in the healthy human gut microbiome. And then I did a screen of 1,200 FDA-approved drugs, and that selection had both antibiotics and human-targeted drugs. I guess you can imagine this is so in the beginning, we just did it at one concentration, which on average, we feel these drugs would reach in a large, intestine. So yeah, I mean, that's one concentration so far. And we did this in our high throughput setting inside an anaerobic chamber. Okay, and this is now summarizing years of work, basically in one slide. So from the drugs that we have tested, yeah, we had some 144 antibiotics, and 77% of them inhibited at least one strain from our selection. But what you can see in the color bar here, in many cases, we had all of the 40 strains inhibited. So antibiotics have a broad impact on gut microbiomes. So that is nice. And in the beginning, I thought, okay, the control works, so I'm happy. But I will tell you a little bit more later on also about the antibiotics. We also found that not only metformin, so metformin was the motivation for us to get started, yeah, because that was such a wow, non-antibiotic drugs can inhibit gut commensals. Is this a broader effect? So that was the motivation. But in the end, what we found is that from more than 800 drugs that we have tested, more than 200 inhibited at least one strain from our selection. So this is rather common that you have a side effect of a drug on the gut microbiome or members of the gut microbiome. And what you can nicely see here is that the effect is not as broad. So in many cases, it's just 10 or less strains from out of the 40 that we have tested that were actually inhibited. We did then a lot of work to convince ourselves that this is reflected in humans. I mean, in the end, it was a test tube in the monoculture. And we compared to data that was available from cohort studies. We find quite high concordance. And we also did a lot of work, and I have no time to show this today, but the effect seems also to be reflected in side effect patterns. So just from the side effect pattern of a drug, we can actually predict whether we would also expect antimicrobial activity against the panel of microbes that we have tested. So if we look into the different therapeutic classes, we find effect across all therapeutic classes. However, they are particularly enriched for the antipsychotics and for the antimetabolites. For the antipsychotics, I think this is quite surprising. Yeah, I mean, the transporters and the receptors they are supposed to target in humans are not necessarily known to be there in bacteria. However, there's a lot of signaling going on along the gut-brain axis. So this could be some drugs where you can also imagine it's part of the mode of action to also, act on the gut microbiome. For the antimetabolites, that makes more sense. It's conserved pathways between pro and eukaryotic cells, and targeting those, of course, would then also have an effect on gut bacteria. Okay, so something what we also observed very early on is an interesting correlation, and that's basically shown here. So each point basically is a species, and on the x-axis you have the number of antibacterial drugs that would inhibit a species, and on the y-axis you have the number of non-antibiotic drugs that would inhibit a species. And overall, there's quite positive correlation, meaning species that are more sensitive to antibiotics are more sensitive to human-targeted drugs. And we wanted to test this. So what we did is we took an E. coli strain. So E. coli is down here. And we reasoned that if we knock out an antibiotic resistant mechanism, the strain should not only be more sensitive to antibiotics, but also to human-targeted drugs. And this is exactly what happens when we knock out Tol-C, which is an antibiotic efflux pump. The strain then is also more sensitive to human-targeted drugs. We find this also in other species. Here, Bacteroides uniformis. In isolate, it's more resistant to antibiotics, also more resistant to human-targeted drugs. Okay, then we wanted to know, how does this work? Can we do this more globally? So what we did here is we took E. coli and we had an overexpression library. So basically we overexpressed each individual gene of the genome of E. coli and asked the questions, which gene do we have to overexpress to rescue E. coli growth in the presence of the human-targeted drug? And the result, so this is the human-targeted drugs that we have tested, and the result is basically, here. So everything that is boxed here is antibiotic-resistant genes. So if you overexpress an antibiotic-resistant gene, E. coli will also become more resistant to antibiotics, to non-antibiotics. And so there's a cross-resistance of antibiotics and human-targeted drugs, which is kind of scary because that implies that there might be a risk of acquiring anti-resistances by consuming non-antibiotic drugs. And by now there are some cohort studies that also have similar results. So that's actually seem to happen in patients as well. So summary so far, I've showed you that there is an intensive impact of drugs on gut microbes, a direct growth effect. And it's not only antibiotics, but also non-antibiotic drugs. This is relevant in vivo. We see this in a side effect pattern. Certain therapeutic classes consistently affect gut microbes, so several members of the drug class affect gut microbes. And we find that antibiotic-resistant mechanisms can also protect against human-targeted drugs. Okay, now that was published in 2018, and we went ahead and actually looked a little bit closer in the antibiotics. So this is the second part. It's also a part of my postdoc work together with Camille Goemans and Nassus Tipper's lab. So for the antibiotics, I mean, we know that there is a strong collateral damage on our gut microbiome. This has long been known. It's more appreciated. So in the recent past, long-term microbial changes in the microbiome composition can then result in the development of allergic, metabolic, immunological, inflammatory diseases. However, we did not have, and I was surprised by myself, we did not have a direct interaction map. So which antibiotic would inhibit which gut commensals. For the pathogens, I mean, this is very well documented, but it was not so clear for the commensals. And we already had in hand the effect of 144 antibiotics from different antibiotic classes on 40 species. And at this concentration of 20 micromolar, and we did like a follow-up, so really concentration resolved minimal inhibitory concentrations that we've tested. Both data sets are in quite good concordance, but of course we can learn way more from the concentration resolved data set. And that expanded the available MICs for commensals by 80%. What was known from commonly available databases. What can we learn from the data? So this is just counting the number of inhibiting strains by certain antibiotic classes. And I think you can appreciate that there are class-dependent effects. So aminoglycosides do not do very much. This is known under anaerobic conditions. So aminoglycosides need to be actively imported, which requires energy levels low in anaerobic conditions. So this was expected. Then we find here fluoroquinolones. Fluoroquinolones are all over the place. However, if you take a closer look, you actually can see that this is generation dependent and newer variants, so newer generation having a stronger impact on gut commensals. Again, that makes sense because the idea was we want to have a broader spectrum so that they are not only inhibiting gram-positive or gram-negative, but both. And that has then, as a consequence, a broader collateral damage on gut commensals. Okay, and then for beta-lactams, again, all over the place, we followed up on 19 Bacteroides and 8 beta-lactam antibiotics. So what you basically can take home from here is that the resistant profile does not at all recapitulate phylogeny, meaning this is really a strain-specific trait. Then macrolides and tetracyclines would just inhibit all of the members that we have tested. Now that was surprising to us because we basically thought, macrolides and tetracyclines are those class where we actually would expect, where we were quite surprised. So again, for the beta-lactams, maybe I start with beta-lactams. Yeah, they are bactericidal antibiotics. We know it's a strain-specific trait. So what makes sense is if you inhibit individual members of the gut microbiome, of course, you have an overall big shift. Now, macrolides and tetracyclines, we know from patient data, yeah, that they have a strong impact on the gut microbiome composition. However, they are textbook bacteriostatic antibiotics. So that means they are just supposed to inhibit, but not necessarily to kill bacteria. However, if you just inhibit but not kill, yeah, and then you inhibit all of them, how can this change the overall composition? And that is something we looked into. So for a handful of different gut microbes, we actually checked the percentage of survival cells after 5 hours of exposure. And what you can nicely see here is that in many cases, we have a drop below a threshold of 0.1%, which is a clear bactericidal activity. So although from pathogens, they are more bacteriostatic antibiotics, they had bactericidal activity on commensals, which you can nicely see. If you look into microscope, you see all sort of shape defects, but also you see lysis here in the growth curve. And we think that this differential bactericidal activity in the end is the reason for the large effects on the microbiome composition. And this is important in the community context. So this is here for E. coli. Here, one E. coli is really, it's a bactericidal activity, it's a bacteriostatic activity, which is also true in the community context. Now, if we treat and we do a washout and treat again, in this case where we have a bactericidal activity, you really lose the E. coli from the community, meaning this is important in the community context where there is strong competition. So you will just get out diluted. Okay, and for the last couple of minutes, I would like to show you another thing that we can nicely do with our setup, and that is find ways to circumvent antibiotic, the collateral damage of antibiotics on gut commensals. So at the time where we started this part of the study, there was another postdoc in the lab that could show that drug-drug interactions are very species-specific in bacteria. And then we thought we can use the species specificity to selectively protect commensals from antibiotics, but the antibiotics should still work on pathogens. So we thought we could find, just by screening, compounds that would selectively antagonize the effects of antibiotics on commensals. So we tested many compounds in our screening setup to find such antidotes that would protect commensals. To put a long story short, we found several ones, and I would like to show or tell you a little bit more about two of them, which is dicumarol and benzbromarol. So what we do, the assays that we do this checkerboard assay. So here's Bacteroides vulgatus. This is the area under the growth curve. Orange means perfect growth. At a certain concentration of erythromycin, the bug cannot grow anymore. However, you can rescue growth if you add benzbromarol and dicumarol. You can also rescue the percentage of surviving cells within the community. And that not only works for Bacteroides, but for a bunch of different other Bacteroides as well. However, it does not work on a pathogen. So Staph aureus, E. faecalis, Faecium, they are just not inhibited. So this is exactly what we were looking for. We also tested this in a community setting. So when we add kind of this antidote, so this is basically the normal composition. This is the erythromycin treated. However, if we add the antidote, we have a composition that very well resembles the untreated community. So we can rescue or we can prevent the collateral damage. We can also do the same in a community where we just spiked in the pathogen. This is basically showing that the community cannot grow any longer. The community can grow in the presence of the antidote. However, we cannot detect any Enterococcus faecalis in those communities any longer. It also works in stool samples. So what we did here, we had the stool from the patient treated in vitro with erythromycin. The size of the circle basically indicates the number of OTUs that were sensitive to erythromycin, which you can see in all the 9 individuals that we've tested. Most of them had reduced OTUs for bacteroidals, clostridials, and others. Those individuals just had resistant bacteroidals, so that happens also. And then, of course, in this case, you cannot protect them because they're already protected. Now, if we basically add dicumarol, and this is now the shade, basically, of these circles, you can see in all cases that we were able to rescue bacteroidals. Overall, we could rescue more than 50% of the bacteroidals, and in many cases it was Bacteroides. So this is exactly what we screened for. We could rescue Bacteroides within these communities. Ultimately, we did the animal experiment. So we had mice that were colonized with a defined community. There was our Bacteroides vulgatus in there. If you treat with erythromycin, there is a decrease or drop in Bacteroides vulgatus level, and then there is basically rescue if you remove like the drug. However, if you have a co-treatment with the antidote, this drop is less dramatic. So there is really a protection of Bacteroides also in the mice. So this is shown here for decumarol, but we find similar effects also for benzbromarone. So it's also possible in an in vivo setup. So to sum up, I think in the second part, I showed you a high resolution map of antibiotic effects on gut microbes. We are now interested more in the strain level pattern because we know in particular for the beta-lactams that this matters. I've showed that macrolide and tetracyclines act bactericidal on gut microbes. This is just very few words left. I'm done in a second. So the consequence on the decline on microbiome diversity is basically what we are interested in. I showed you the antidotes as a concept to selectively protect gut microbes under antibiotic treatment. Here we are curious about the underlying mechanism and whether we can use those antidotes also for other microbiome-related diseases. And with this, I would like to thank my group here in Tübingen, a lot of collaboration partners, our funding. Thank you for your attention. And I hope I could have showed you that you can also take approaches to the microbiome field that it's not necessarily based on metagenomic sequencing. And yeah, I'm also looking forward to hear about your questions in the panel discussion. Thank you.